Hello everyone, welcome back. We're still talking about the development of spinal cord. In this section, we'll see the neural crest cells as well as the ectodermal placodes. These are the other two important segments that are given off by the neuroectoderm. The first being the neural tube, which has already been discussed in the first section. In this section, we'll see how the formation of the neural crest cells takes place. Then we'll see what are the ectodermal placodes, how they're incorporated in the neuroectoderm, and what adult structures do they give rise to. And finally, we'll talk about the clinical correlates as to what happens when the developmental processes do not go as planned. So, getting into it, first of all, we have the formation of the neural crest cells. I like to take you back again to the process of neurulation. So the process of neurulation takes place in a an early 3-week embryo, which is at a 3-layer stage. The process of neurulation is actually taking place in the uppermost layer, which is the ectoderm. The ectoderm thickens to form the neuroectoderm. This neuroectoderm is separated from the rest of the ectoderm by the neuroectodermal barriers or neuroectodermal clefts. These are known as the neural crest cells. So let's see the neural folds. These are the neural folds. They come together and fuse. These are the neural folds. These are coming together and they're converging upon each other to fuse with each other. Now cells at the tip, these light green cells, they break away from the neuroectoderm to form the neural crest cells. So these cells actually break away before the fusion of the neural folds to form the neural tube. And these cells are known as the neural crest cells. The surface ectoderm of one side becomes continuous with the surface ectoderm of the other side over the neural tube, as it is pretty obvious since there's convergence of the neural folds, so the surface ectoderm of both the sides become continuous with each other. Now, the neural here you can see the neural crest cells come to lie just above this neural tube, hence they do not participate in the formation of the neural tube. At first they remain in the midline between the dorsal surface of the neural tube and the surface ectoderm. Here you can see they are lying in the midline of the dorsal between the dorsal surface of the neural tube and the surface ectoderm. Now they form two cell clusters dorsolaterally, one on either side of the neural tube. With development, there are two clusters that are formed on either side of the neural tube. They are the dorsolateral clusters. Now the neural crest cells differentiate into cells of the dorsal root ganglia. So these are the neural crest cells. These are the dorsolateral thickenings that are formed by the neural crest cells, and they give rise to a variety of cells. Here you can see all of these cells are given off by the neural crest cells. So among the cells that are given off by the neural crest cells are the cells of the dorsal root ganglia. The sensory ganglia of the cranial nerves are also formed by the neural crest cells. The autonomic ganglia, here you can see these are the autonomic ganglia. Now the cells of the adrenal medulla, here you can see cells of the adrenal medulla being given off. The chromaffin cells, the chromaffin tissue, the melanocytes, the Schwann cells. So all of these cells are actually, the neural crest cells are precursors of these cells, and they give rise to all of these adult structures. So moving on, what are the ectodermal placodes? So prior to the neural tube closure, before the closure of the neural tube takes place, here in the figure you can see the neural fold contains the neural crest cells, and these are the neuroepithelial cells. So now the neural crest cells are detached. Here you can see the neural crest cells have been detached. These are the detached neural crest cells, and the neuroepithelial cell get incorporated into the surface ectoderm. Here you can see these in blue were the neuroectodermal cells. They get incorporated into the surface ectoderm. They do not become a part of the developing neural tube. However, they become incorporated into the surface ectoderm. Now, the areas of neuroepithelium within the surface ectoderm are termed as the ectodermal placodes. So, ectodermal placodes are actually the areas of neural tissue or neural epithelium in the ectoderm after the formation of a neural tube. So, the neural tube consists of the neural crest cells and these neuroepithelial cells. These neuroepithelial cells, they do not become a part of the neural tube but become a part of the overlying ectoderm. These neuroepithelial cells are now known as the ectodermal placodes. So, moving on to the clinical aspects of this development of the neural tube, we have anencephaly. Anencephaly is a condition in which there's failure of the cephalic part of the neural tube to close, and there's associated defective development of the vault of the skull. So, we talked about it earlier as well. The anterior neuropore, it closes at early 4th week of development at about 20 somite stage. So if there's failure of the closure of cephalic part of the neural tube, it is also associated with the faulty development of skull. It gives rise to anencephaly as shown in the figure. So vault of the skull is absent. Here you can see the skull vault is completely absent. Now, brain is represented by a mass of degenerated tissue. This is the mass of degenerated tissue which was supposed to be the fetal brain. This degenerated tissue is exposed to the surface, and this tissue undergoes degeneration because it is exposed to the amniotic fluid, and amniotic fluid is actually dangerous to the developing brain. Now, cord is open in the cervical region. Obviously, we talked about it before. This is due to defect of the failure of closure of the defect or failure of closure of the anterior neuropore. So we have prominent eyes. These are the prominent eyes which are bulging forward. Chin is continuous with the neck due to chin is continuous with the chest due to absence of the neck. So there is little or no neck, and chin is almost continuous with the chest. Now, moving on, we have Rachischisis. So, in Rachischisis, there's incomplete closure of the caudal neuropore. So, initially, anencephaly was incomplete closure of the or lack of closure of the anterior neuropore. In Rachischisis, we have incomplete closure. There's somewhat closure, but not a complete closure of the caudal neuropore, and there is defective development of associated vertebral arches, and it causes a congenital anomaly. So it is present at birth. So there are various conditions that can occur besides Rachischisis. These are also congenital conditions. This is spina bifida occulta, in which only a small defect, a skin defect, is actually present. The central nervous system, the spinal cord is actually intact, as it is not being exposed to the exterior. In meningocele, we see the subarachnoid space being extended into the lesion. We can see the meningeal component being extended into the lesion, and there's a protruding part from the lower back of the patient. In the C figure, we can see meningomyelocele, in which both the meninges as well as the neural component of the spinal cord, it herniates outside or it herniates into a small closed sac which protrudes from the dorsal body wall. Now, this is Rachischisis. In Rachischisis, this is incomplete closure of the posterior abdominal, incomplete closure of the caudal neuropore, as well as the defective development of associated vertebral arches, as a result of which the neural tissue may be folded upon itself, or the neural tissue may be normal, but it might be exposed to the environment in the form of a protruding bulge from the lower back. Here, this you can see this is Rachischisis, is failure of the dorsal portion of the vertebral arches to fuse with each other. Here you can see. So the dorsal portion of the vertebral arches, they fail to fuse with each other, as a result of which there's a swelling in the lumbosacral region. The neural tissue is widely exposed to surface, and sometimes it can show considerable overgrowth in the fetus. However, this exposed neural tissue is usually not in a very good condition, because amniotic fluid is not conducive for the development of this spinal or this brain matter. So this was all about the clinical correlates of the neural tube development. In this section, we talked about the neural tube, the clinical correlates of the neural tube. We also saw what ectodermal placodes were and how neural crest cells developed. These are important concepts. You have to understand them and memorize them. So I suggest to take a copy and a pencil and try to write it down, so that you can understand what happened and how it happened. For further sections, keep watching sqadia.com.